Hi, I'm Dr. Marc Rothenberg here at Cincinnati Children's Hospital, and I'm very pleased to share with you the results of our recently published paper in Science's Signaling. In this paper entitled TSLP Shapes the Pathogenic Responses of Memory CD4 Positive T Cells in Eosinophilic Esophagitis, we report on critical information concerning this cytokine and the disease. TSLP, which is abbreviation for thymic stromal lymphopoietin, is a cytokine that's known to induce type 2 immune responses, primarily by targeting the development of memory pathogenic effector T cells. Few studies have examined the effect of TSLP directly on the pathogenic cells rather than their development. In fact, most studies concerning the effects of TSLP on T cells have been conducted in mice. In our study, we investigated whether TSLP contributes to allergic inflammation by directly stimulating human CD4 positive T cells, and whether this process is operational in eosinophilic esophagitis, a disease linked to variants in TSLP. We show that about 10% of esophageal-derived CD4 positive T cells from subjects with EOE and less than 3% from control individuals expressed the receptor for TSLP and directly responded to TSLP as measured by the phosphorylation of STAT5, a transcription factor activated downstream from the TSLP receptor. Accordingly, increased number of TSLP-responsive memory T cells were present in the circulation of subjects with EOE. Furthermore, TSLP increased the proliferation of CD4 positive T cells, enhanced type 2 cytokine production, and induced an increased abundance of its own receptor, and also modified expression of 212 genes. The epigenetic response to TSLP was associated with an enrichment in BATF and IRF4 chromatin binding sites. And in fact, these transcription factors were induced directly by TSLP, providing a feed-forward loop. The number of circulating and esophageal CD4 positive T cells in these patients correlated with the levels of eosinophils in the esophageal biopsies. These findings provide the basis for a blood-based diagnostic test based on the extent of TSLP-induced STAT5 phosphorylation. These findings also provide the rationale for looking at anti-TSLP-based therapeutics for the treatment of EOE. This data is summarized in a graphical abstract, which I will show you. One can see in this figure that TSLP binds to its receptor expressed by pathogenic effector T cells. TSLP induces phosphorylation of STAT5. This leads to epigenetic changes, particularly those driven by BATF and IRF4 binding to DNA. This leads to T cell stimulation, which includes release of critical cytokines such as GM-CSF, which activates dendritic cells to further polarize Th2 cells, as well as the stimulation of them. These activated TSLP-stimulated cells also release relevant cytokines that directly target Th2 cells, particularly IL-2 and IL-4, and the pathogenic cells stimulated by TSLP release increased amounts of type 2 cytokines, mainly IL-5 and IL-13, as well as IL-4 and IL-3. IL-13 directly targets epithelial cells, which make TSLP, and IL-5 and IL-3 target mast cells and eosinophils, and this leads to enhancement of the allergic inflammation. So you can see this pathway showing multiple effects of TSLP at the level of the pathogenic effector cells, which drive eosinophilic esophagitis and other allergic diseases. These findings really provide the rationale for continuing to pursue anti-TSLP-based therapeutics for this disease and others. Thank you very much for your attention. I hope that you will enjoy reading the manuscript.